Okay, well, welcome back. We'll get started now. This is Module 2.1. I'm going to be talking about contact energies and cell sorting today. I hope that we feel that this is a natural continuation of the things that we were doing yesterday afternoon. Hayden will post the various links in the chat for downloads and for the Slack and for the hackathon. Again, this meeting is live streamed and recorded, so please be aware of that. And I also want to thank our funding sources from the NIH and NSF. And I should say that many of the slides we're going to be showing here were developed by Giulio Belmonte, who was speaking yesterday. And so I want to thank him for making those available. So today we're going to be talking about contact energy. We heard about what are called plugins, which specify energies and forces on cells in CompuCell. The form of the constraints that you're going to be hearing about a little later today are a little bit different from the contact energy. And so I'll talk a few minutes about that. We're going to talk a little bit about some biology of cell shape, although we're not going to go into great detail on that. A little bit about the epithelial to mesenchymal transition, which is important in cancer progression. And then we are going to simulate an aggregate of cells, explore what the contact energy does to that aggregate of cells, and then look a little bit about what happens when you do the same thing for a single cell and some of the pathologies that you can get in that situation. I hope we'll talk a little bit about contact energy neighbor order, and then we'll introduce one of the classic simulations, which is cell sorting. And I will begin that, and Hayden may pick up and do a little bit more on that as well. There may be some problems that we don't get to today, which you can think of as homework assignments, in which you can do a little bit more exploration. I don't want to take too much time, but these are fundamental concepts, and we need to make sure that everybody is comfortable with them. Some of them seem very natural. Some of them are a little bit counterintuitive, and feel free to ask in the chat any questions you have about these basic ideas. I should say that there's going to be some mathematics here. The basic ideas are rather simple. You don't need to follow the detailed mathematics to be able to work with these ideas. Fundamentally, we're thinking about how things stick to each other. And the idea is that some things stick to each other more strongly and some things stick to each other more weakly. Some things push each other away. The detailed mathematics of how that's implemented is helpful if you're going to do advanced work, but it's not absolutely necessary to understand the math to be able to follow the basic ideas. So back in module 1.3 yesterday, when Giulio did his introduction, we talked about energy constraints, and you heard about the volume, surface, and length constraint. And Gilberto in the next module is going to be doing exercises on those in some detail. Those all have the structure of what are called constraints. That is, there is a target value, a property, a target value, and that target value is compared to the existing actual value of that. For example, target volume of 10 and an actual volume of 6, target volume of 10 and an actual volume of 14. You can assign that units if you like, microns cubed, centimeters cubed. And we take the difference between the two. If they're the same, the difference is 0. And we square it, because what we want to do is we want to have a penalty when we move away from the target. We don't care whether we're moving up or down. We want to have the penalty be 0 when we're at the target and large when we move away. And there's a lambda volume, lambda surface. That lambda is the strength of the constraint. And you can think about a spring that's pulling you back to a given position. Lambda is the strength of the spring constant. The equilibrium length of the spring would be the target value. And so all of those constraints act to move the system back to closer to the target. The contact energy works a little bit differently. It is not in the form of a constraint. And that means that it has some features that are somewhat different. It's quite important. Contact energies determine how smooth or rough the surface of a cell is. It determines whether cells of a given type tend to condense with each other, and we'll do that in a few minutes, or spread out in medium. It determines whether cells of different types like to stick to each other or don't. And it'll show us to see that it determines the spatial pattern of organization of tissues. And that actually is one of the very fundamental principles, both in developmental biology and wound healing. And its failure is one of the causes of metastasis in cancer. So one thing that we want to think about, and I'm going to skip these slides a little bit quickly. As I say, this is Giulio's deck. He likes to talk about it a lot, is that cells have a lot of different shapes. There are two fundamental classes of cells, although there are a lot of complex shapes and intermediates. There are what are called epithelial cells that stick closely to each other and form sheets. And there are what are called mesenchymal cells, like fibroblasts, which have more disordered structures and form, in particular, connective tissue. There also are cells that have very specialized shapes, like a sperm cell or a nerve, that don't really fall into either category, and we're going to neglect those for the moment. You can have fairly detailed cell shapes in CompuCell, but you're not going to be building a Purkinje neuron in CompuCell. Suppose you could, but it would be an expensive thing to do. But the key idea that we're going to be thinking about now, and it's going to come back at the end of the course, where we're going to teach you how to build models of epithelia that are much more detailed, are epithelial structures versus mesenchymal structures. And today, the only thing we're going to be thinking about is that in epithelia, there are adhesions that bind the cells closely together. And in mesenchyme, the cells are often in contact with each other, but more loosely connected. There are a whole bunch of different properties that distinguish epithelial from mesenchymal cells. And there are a lot of intermediate states as well. But for today, we're going to neglect really everything except the fact that if the cells are tightly coupled to each other, we're going to think of them as epithelial. And if they're loosely coupled, we're going to think of them as mesenchymal. There definitely are particular molecular markers and also cytoskeletal architecture that are different. And those will be dealt with later in the week in detail. During development, you see switching very often between epithelial structures and mesenchymal structures. When cells are going to form adult tissue, typically in layers, they're going to form epithelia. On the other hand, when tissues have to reorganize, the junctions between the cells have to loosen so that cells can move relative to each other. It's not impossible for cells to move within epithelia, but they're relatively limited. So it's pretty often that you'll see something like this in a normal development, where you have primitive epithelium, for example, before gastrulation. You have mesenchymal phenotype during gastrulation, and then you form a new epithelium from the same cells. So cells go back and forth between being adhered to each other and being more loosely coupled to each other. And this is something that happens pretty often in cancer. In particular, most cancers, not all, but most cancers are carcinomas. That is, they're derived from epithelial cells. Some cell cancers are sarcomas derived from mesenchymal cells. But one of the key hallmarks of cancer is the loss of epithelial organization toward to have a more mesenchymal phenotype. And in metastasis, typically the cells that are migrating have a mesenchymal phenotype, although they may reassume an epithelial phenotype and the secondary site of metastasis. And so this is just a statement again about this transition from a typical epithelium to a disorganized epithelium to an invasive phenotype in a frank cancer, then potentially migration through the bloodstream, although that's certainly not the only way cancers metastasize, the formation of a secondary tumor. You already saw yesterday in the short talk that was given by Joel that you can have disorganization of epithelia in the eye after injury. Jim Sluka is going to give a breakout session where he talks about epithelial disorganization in ovarian cancer. And so epithelial disorganization is a key hallmark of cancer. I'm not going to go into the particular molecular details here. These are going to serve as a reference. One thing to be aware of, though, is that there is a continuum between epithelial and mesenchymal phenotypes. So I'd like to build a very simple simulation now. Avishek, what's up?
Yeah, so I was wondering in the previous slide, what does the EMT and the MET stand for?
EMT is epithelial to mesenchymal transition, and
Okay.
MET would be mesenchymal to epithelial transition. So
Okay.
you can go both ways.
Okay.
I always find it a little bit confusing because I would think there should be a lowercase t, MTE, mesenchymal to epithelial, but the abbreviation has the T
Yeah.
at the end of transition.
Okay. Thank you.
Okay. So I would like everybody to launch Twittit. Please remember how do you launch Twittit. And I'd like a hands up when you have Twittit running. And we're going to build a simple simulation together. And I guess I can ask you as a show of hands, perhaps, would you prefer I do a screenshot and code with you? I think it's a little hard for people to see the screen if I make it small so that you, I think we could go through it again. I think what I'll do is I'll walk you through the steps of building the simulation, and then perhaps I can build it in front of you. It only takes a few minutes to do. So I'd like everybody to be sure that they have Twittit running. And this should be a review at this point, although I realize that it takes time to get familiar with using Twittit. So please get Twittit running. Okay. I'm going to do one of those completion polls just to make sure that we're all on the same page here. Realize that the poll pops up on your screen, but want to make sure everybody's got Twittit running. Somebody says they need help. If you need help, please reach out to one of the TAs and join the breakout room. Would the person who needs help please raise their hand or put in the chat their identity so we can get you help? Okay. So let's do what we did yesterday. We're going to build a simple simulation. And what I'd like you to do is go to CC3D project, new CC3D project. Right? And now you'll pop up CompuCell Simulation Wizard. and you are going to browse to a site where you can find someplace you can write to, and type the name of the simulation. I called it module 2_1_1_EMT. However, you can use any name you like as long as there are no spaces, dots, slashes. Don't start with a number. And I realized sometimes some of the screenshots may not have the same name in them because they're screenshotted from different versions of the course. Don't be put off if you see a picture like this and inside of simulation name it has a different name. Any name that you use can be all right as long as it follows the rules for naming. Okay, hit next. And now the next screen, you shouldn't have to do anything. Rectangular slab, lattice dimensions, boundary conditions, no flux, lattice type square, average membrane fluctuation 10, pixel copy range 1, number of monocolor steps 10,000. Those numbers should now be beginning to mean something to you from the things you've done in the past day. Okay. Hit next. And now you're only going to create one cell type. So type the name of a cell. You could type my cell, cell 1, no space in the name, no special characters in the name. And hit add. And after you've added that one cell type, we're only going to work with one cell type, hit next. Okay. We're not going to use a chemical field, so hit next. Okay. And now you should be at the cell property and behavior window. Does anybody need more time to get here? Or need me to review the steps? If you do, please say something in the chat. If I don't see something in the chat, I will assume that you're okay. If you have trouble, we'll be able to download the code from the website if you need it. We are going to use two plugins. Back one step. Okay, let's walk through this again. Twit it, new CC3D project. Type a name for the file. Hit next. Skip this, so hit next. Type a name of a cell type. Cell is fine. My cell, cell 1, anything like that is fine. Hit next. Ignore chemical fields, hit next. And now in the cell properties and behaviors, select contact, volume flex. That's all you have to do. There's a question between volume flex and volume local flex. Volume flex defines the volume of all cells of a given type, and it does it in XML. Volume local flex allows you to have each cell have its own volume and to change the volume of the cell one at a time, but it means that you have to define the initial volumes in Python. And so today we're not going to be using volume local flex until we get to cell growth. Good question. Hit next. And now hit finish. So again, the steps are outlined here. So everybody got through that okay? Okay. Everybody's okay. And now you have to remember how to open the simulation. To open the simulation, click on the name of the file here in Twit, and then right-click and select Open in Player. And Player should open for you. And you should see the name of your simulation in Player. There's a question about the difference between contact and contact internal. we're not going to be using contact internal till later in the class. Contact internal is only for compartmental cells. Good question. Okay. Does everybody have this open in Player? Does anybody need help with that? Okay. That's a great way to tell us that it works, is to put a screenshot of it in the chat. Okay, Avishek says you need help. Player is not opening. Okay, Yaren says they need help. When you say player doesn't open, could you type in the chat what is happening? Can you launch player separately instead of launching it from Twit? You can certainly close player first if you like. That would work. You can also hit stop inside of player and open the file that you just saved. That would work as well. Anybody else? Again, if you're stuck, please help each other in the chat. And again, if you're stuck, please reach out to one of the TAs for help. Okay, so people should be seeing something that looks like this. Remember the stop function. Stop terminates the simulation, the pause button, the single step button, and the play button. I'll give people a minute to catch up. Again, if you need help, reach out in the chat. Does anybody else need help at this point? There was a question earlier saying we should have homework. We don't like to assign homework. You're working hard enough. But if people have had issues, for example, you haven't understood well how to use a player or Twit, or you've had trouble opening them, then it's important that you practice on your own after the class is over or in the morning before class, so that you make sure that everything works. We definitely want to support you in the class time, but it's easy to get behind, and so it's important to try those things out as possible, if possible, on your own. Good. Looks like Kimberly has it running. So could TAs please take care of those questions in the chat? That would be very helpful. Thank you. Okay. So one of the things that we need to think about is when we do simulations is describing what you see in a simulation. And that may seem like an obvious thing to do, but there are a lot of features that you might address in a simulation. And some of these may be things that are visible in a single screenshot. Some of these things have to do with how things change in time. And there are a couple of basic principles that we could think about. One is, what is the overall shape of the tissue? Is it square? Is it round? Does it look like a stop sign? Is it an octagon? Is it rough on the surface or smooth on the surface? Those are all questions that we could ask, and they all exist in biology in one form or another. And it's important to train ourselves to look for important features in simulations. We could also zoom in and look at the individual cells. Our initial cells in our simulation, because of the way we set it up, are square. That's not very realistic. As time goes on, those cells will become more rounded or more hexagonal. And again, we can look at the boundaries and ask, are those boundaries smooth or rough? And are those boundaries lined up with the lattice axes, which is almost always unrealistic, or are they basically independent of the lattice configuration? We can also ask, how fast do changes happen? Some things, like the boundaries between the cells, change very rapidly. Other things, like the overall shape of the aggregate, change very slowly. So you should ask macroscopic questions about shape and organization, microscopic questions about shapes of cells and their organization, and always ask what the time scales for things are. Are things happening fast or slow? Abhishek.
Yeah, so I'm a little confused about the independence of this with the lattice configuration. What does that mean?
So the lattice is square.
Yes.
In reality, the real world doesn't have a lattice in it.
Uh-huh.
And therefore, if you see things lining up along the lattice directions, you're seeing something that reflects the lattice geometry and not the biology.
Okay.
And we have plenty of time to see what is a good simulation in that sense and what is a bad one. Um,
Okay.
in computing, you always have to discretize things. There is no continuum in computing.
Yeah.
You either discretize things on a lattice, the way we're doing it here, which has the great advantage of being very simple computationally, but has some artifacts associated with the fact that there's a lattice. Or you do things like finite element methods, uh, which are off lattice, but are computationally more expensive. And so there's a trade-off always between those two
Yeah.
things. But I remember many years ago hearing a talk where somebody was observing animals in the wild, and they discovered a pattern of a weekly periodicity of something. And the answer is that the observer is making a mistake. The person who was doing the observations on Sunday was different from the ones on the other days, and they were doing the observations
Oh,
differently because animals don't know about the days of the week.
yeah.
They know
Okay.
about days, they may know about months, but they
Uh-huh.
certainly don't know about weeks. And so if you see things lining up in a direction along the lattice, that means that your simulation is reflecting something about the simulation methodology and not the biology. Okay,
Oh,
we'll come back to that
yes.
as we go
Thank you.
forward. We talk about pathologies of these simulations. Great question.
Thank you.
Okay, so does that work for people? I think everybody hope is caught up there. And now we're going to talk about where this is coming from. And we're going to talk about the contact energy plugin. And the contact energy plugin is one of the most used ones in CompuCell. Um, it's actually more of a physicsy concept than a biological concept. Um, and if you're not familiar with vector notation or summation notation, don't be frightened. The basic idea here is the following. We are going to define a contact energy per unit area between every pair of cell types in our system. So if we have three cell types, A, B, and C, there's going to be a contact energy per unit area between A and A, between a cell of type A and B, between a cell of type A and C, between a cell of type B and B, of B and C, and between C and C. If I have two objects and I bring them in contact with each other, there is some adhesion between them or some repulsion between them. If the contact energy is high, they repel each other. If the contact energy is low or negative, they adhere to each other. So that is the fundamental concept that we're going to deal with here. If we are on a lattice, we can represent this mathematically by saying that we are going to walk over every boundary pixel of a cell and look at all of its neighbors and add up the adhesion energy per unit area with the neighbor. And this summation notation is simply saying we're adding up all of the boundaries and all of the contacts between every cell and its neighbors. Something that is going to come up a little bit later is that there's going to be an interaction range. we don't have to add up only between the nearest neighbors. And the reason for that, again, is going to come up with this issue of lattice anisotropy. It's not something you have to worry about if it's unfamiliar to you. It's a physics question, and you need to think about this a little bit. There's a question about trying to correct for lattice anisotropy, and there are programming methods that can do that. If somebody would like in the hackathon to write a corrected contact energy form, we'd be delighted to have them do that. The Morpheus package has the lattice corrections built into it, and we've thought about implementing those in CompuCell. But the one we're working with today doesn't have those corrections built in. So the first sum is over all cells. So we have to sum up the lattice energy for each cell. And then for each cell, we have to walk around its perimeter. That's why there are two summations. The key thing to remember here, and this is perhaps a little counterintuitive, is that larger energies mean less adhesion. So if I have a big energy of contact, that means I repel. A smaller negative contact energy means I attract. And that may be a little bit backwards from what you expect. Avishek, did you have another question?
Yes, I'm
Go ahead.
sorry for interrupting so often. So, I see that the matrix J is upper triangular. That means that it is symmetric, right? So,
Yes.
is it true that the interaction energy between A and B is the same as the interaction energy between B and A? Like, what I'm trying to understand is like a cell type A has a specific bonding affinity towards a cell type B, but the cell type B might have a different kind of like bonding affinity towards A.
No, that's not possible in this case, because you think of two cells coming together and they
Mhm.
touch, and the energy is an energy, a joint energy between both, and so it's always symmetrical.
Oh,
There
is
are
a
other
joint.
signals, there are other signals that are going to be asymmetrical. But contact energy, if you think about two particles coupled with a spring, the force
Mhm.
from left to right has to balance the force from right to left. Otherwise, the thing would move.
Okay.
So
Yeah.
it
Okay.
is in fact obligatorily symmetrical. That's a good physics question. Thank you for catching.
Okay. Okay. Thank you.
Thank you. Okay. Other questions at this point? Voxels and pixels. Uh, it's a 3D simulation, so in principle, they're voxels rather than pixels. Okay. Something that we have to be aware of a little bit, we haven't really talked about the lattice very much. The lattice in CompuCell can either be 2 dimensions or 3 dimensions, and you have an option of a square lattice or a hexagonal lattice. And actually, using simulations on hexagonal lattice is actually nicer in many ways than square lattice. But it's a little more complicated to think about things, so we're going to typically do exercises here in square lattice. People can easily switch that in the XML if they want to explore the difference. We need to think about neighbor order. If we have a voxel, its nearest neighbors are up, down, left, and right. It has 4 nearest neighbors in 2 dimensions. The second neighbors are the corners, which are labeled 2 in this picture. The third neighbors would be going out 2 steps up, 2 steps down, 2 steps left, and 2 steps right. And the fourth neighbors fill in those corners. You can see that if you go out to a long range, this gets more and more like a circle. If you come in very close, you have a square. When you get to fourth neighbor, things get to be a little bit smoother. And so very often we're going to use fourth neighbor interactions. There's a question about computational cost between square and hexagonal lattice. No, actually, computationally implementing a hexagonal lattice is quite easy. For most things, there are a few things in CompuCell that don't work on hexagonal lattices. You could, in principle, have a triangular lattice, but they have undesirable properties. Somebody says triangular and hexagonal can be the same. It's true if you want to think of it that way. It depends on whether you look at your neighborhood or the reciprocal lattice. If I do a hexagonal lattice, I have 6 nearest neighbor sites. And then as I go out, I have 1, 2, 3, 4, 5, 6 second neighbors. And I also have 1, 2, 3, 4, 5, 6 third neighbors. And so the number of neighbors increases as I go to longer ranges. And if you change the interaction range, you have to keep track of that by hand. CompuCell doesn't divide automatically for you. That's something we've talked about implementing, but it's not implemented yet. In general, longer interaction ranges, 3 or 4 in square lattice, 2 in hexagonal lattice, are going to be a little bit better for the simulation. The question about does CompuCell automatically use 4 nearest neighbors? The answer is you have to tell it what range to use. It's a setting. In the very first panel in Wizard, it asks you what range to use for the POTS update algorithm and for the contact energy. There's a question of what range to use as well. I'm not going to go through this exercise in great detail, but in the slides, you'll find a little worked out exercise in which you can calculate the contact energy for a cell by hand. You have to iterate over all voxels in the cell. For each voxel, you find all of the neighbors within the given interaction range that are different, and then you add up the J values. And so here I have a picture with 8 cells in it. We have 3 kinds of cells by color. Here, 3 and 4. Actually, here they're named. They're not. The colors are by cell ID, not by type. So we've said cell 3 and 4 are type A, cells 1 and 5 are type B, cells 2 and 6 are type C, and so on. And we can pick a voxel. Here's a voxel inside of cell 3. Its nearest neighbors are these 4 voxels. It has 2 nearest neighbors that are not of the same cell in the same cell. We look up the cell type. Cell 3 is type A, cell 6 is type C, so we have boundaries between type 3 and type 6. And we have to count up how many mismatched voxels we have. And so we add JAC twice, 3 to 6, 3 to 6. So we have 2 units of JAC. And then we move to the next one, 3. It's next to a 5. And so we get a J, and 5 is of type B, so we get a JAB. And you can keep doing that. Here I've gone to second neighbor, so I've got an extra neighbor for that first voxel, another neighbor from 6, and so I get another JAC, and so on. And so you can try that out by hand if you want to figure out how this works in detail. You don't have to know this, but if you have certain kinds of pathologies in these models, you can do this kind of analysis to see what's going wrong. So again, the nearest neighbor is left, right, up, down. The second neighbor is the diagonals. So if I've defined nearest neighbor interaction, I only go left, right, up, down. If I've defined second neighbor, I do the first order diagonals. If I've got third neighbor, then I go to the second in the up-down direction. If I go to fourth neighbor, I do those corners as well. Okay. I'll make a general mathematical comment that And again, if you're used to the math, this is fine. If the math is unfamiliar, please don't be intimidated. You don't have to know this to be able to do these things. Which is that we are used to what is called scaling. If I have a square and its side length is L, its area is L squared and its perimeter is 4 times L. If I have a circle, its area is pi over 4d squared, and its perimeter is pi d. And I can calculate what's called a perimeter area ratio, where I take the perimeter and divide by the square root of the area, and that characterizes the shape of the object. A bigger number means the object is more irregularly shaped. A smaller number means the object is closer to a circle. And similarly, in 3D, I can define for a cube, the volume is L cubed, the surface area is 6, and there's a missing, well, this is actually, I should be careful here. This should be L squared. There's a missing L squared there. And this, okay. For a circle, pi over 6d cubed, and the surface area pi d squared. So this L got missed from the last one. If I take the surface to volume ratio, perimeter divided by area to the 1/2 in 2D, surface area to the 1/2 divided by volume to the 1/3 in 3D, that gives me a ratio. In the real world, those ratios depend on the shape only and not on the size. On a lattice, you will find that as you change the size of the object, the ratio changes even for the same shape. And that is an issue that we face with lattice calculations. Don't have to worry about it in too much detail now, but again, there's certain pathologies you can run into in CompuCell that it's good to be aware of that. So let me talk about the contact energy and steering. We've already done some steering, but I want to come back to that very quickly. In our little simulation here, why don't people open up TwidIt, go to the XML, and find the contact energy plugin. If you can't do that, don't worry about it. I'll show you on the screen. But you'll see here is our definition. We have 2 cell types, medium and cell. J medium doesn't do anything because there's only one medium cell, so there never is a contact between medium and medium. J cell medium, J cell cell. And by default, you built your simulation with the value of 10 for each of these. If I look in the plugin contact, you'll see J energy medium 10. That doesn't do anything. It's just there to make the lattice symmetric. Medium my cell 10, my cell 10, and the default neighbor order is 4. Okay. You can change these values using the model editor as the simulation runs by opening up. It would be nice if they were open already because it's a little hard to see. If you open up contact and then energy, you'll see 10 medium, 10 my cell medium, 10 my cell medium. And you can type in a new value here. And one trick that you're going to run into if you're typing in the steering panel, in the model editor panel, is that if you see an integer 20, you have to type in integer 10, 5, 4. If you see a float 20.0, you have to type a float 15.5, 15.0. If you type a float when there's an integer here, it will reject it and not change the value. And if you type an integer when there's a float, it will reject it and not type the value. So even if you want to type 5, remember to type 5.0 if the thing that was in there was 20.0. Okay. These are the types of cells. We have cell type medium, cell type my cell, and We've just given them a number, type 1 and type 2. You can refer to cell types either by name or by number. If you want to be more biological and specify contact energies not by cell type, but actually by the molecular species on the surface of the cell, there is a plugin called Adhesion Flex, where you can say, I have a certain number of cadherins of a particular type on each cell, and the contact energy per unit area is such and such based on the number of cadherins. That's very useful. It's more biological. It's also a little bit more complicated to work through, and so we're going to work with this simpler formalism for today. I encourage people always to look at the manuals. The manuals have a lot of detail, and so it's a good idea to read up on this in the manuals. You can also ask ChatGPT. It actually is pretty good at answering questions about these things. Omar, question?
Yes, hello. Is there a relationship between those two kinds of adhesion? So the chemical one and the unit per area?
So in our case, what we want to define is the adhesion energy between two cells. And in one case, we are simply asserting that the cells have some set of properties so that they adhere in a particular way. That's how we're doing it here. We're not asking the question about the details of the molecular forces at work. In Adhesion Flex, we actually define molecular species and the adhesion between them. And so we have more flexibility. We can have much more elaborate kinds of adhesion and dependencies. So for example, if we wanted to model a cell mutating during the progression to cancer, so individual cells had different adhesivities cell by cell, we would have to use the Adhesion Flex plugin, because that would allow us to say this cell has more integrins and fewer cadherins, and therefore it's going to stick to medium more and less to other cell types. And we can do that on a cell-by-cell basis. And Gilberto in the past has given a nice lecture on that. I don't know if we'll have time to do that this year.
Okay, thank you.
Okay, just reminding you, steering in player, only the XML-defined parameters are accessible through the model editor window. When you get to Python ones, you'll have to use the steering panels, the sliders instead. Another problem with editing things in the player window is that it doesn't log the changes that you've made. We've worked on doing that. So if you find a really nice set of parameters, write them down, because you won't remember. If you don't remember them, it's not logged. All right. So let's actually now work with this a little bit. We have two cell types, medium and micelle. Two cell energies, cell medium and cell micelle. Sorry, micelle. Remember, as it was talked about before, JAB is equal to JBA, so we don't need to write that bottom corner. And there's never medium doesn't do anything. So we have two energies, the energy per unit length between cell and medium, and the energy per unit length between cell and cell. Okay. Contact energies set contact preferences. The lower the contact energy, the more the adhesion between two cells. The higher the contact energy, the less cohesion there is. There is a factor of 2 with medium that you have to worry about. And so if J micelle medium is less than J micelle over 2, then cells prefer to stick to medium than to each other. If J micelle medium is bigger than J micelle over 2, then cells prefer to stick to each other rather than medium. And there's a reason for the factor of 2. The reason for the factor of 2 is, suppose that I have two cells that are touching each other, and I have 1 unit of contact energy, 1 unit of area, contact area. Imagine my hands as being 1 unit of area. If I pull them apart and they're now touching medium, I have 2 units of contact because each cell separately is touching medium. And that's why there's a factor of 2. We're starting out with these all being 10 and 10. Okay, so the first exercise in your simulation, and you can do this in the steering panel, is increase the contact energy between medium and cells from 10 to 20. What do you think is going to happen when you do that? Are cells going to stick more to medium or less to medium when I increase the cell medium contact energy?
Maybe increase more, they contact more, the cells.
So higher contact energy means less adhesion. So if I increase the contact energy between medium and cells, the cells will repel the medium more. And somebody says the cells will bind more tightly together. Great. So that's a good prediction. Why don't people try it out? Now, I have a little bit here, which we could skip over quickly. I'm just going to show this to you. I'm not going to make you do it here. You can do 2 things. You could go in to the code itself. This is really more for reference than anything else. Go to the XML. Go to the lines in the XML where we specify the energies. And you could edit that line, 20. And then after you've edited that line, you save the simulation. Save all, I always use. The double floppy disk icon or save all here. And then if you stop the simulation in Player and restart it, it will load the new values. Player will automatically, when you hit stop, check to see if the simulation has been updated and updated for you. That's one way to do it. The other way to do it is steering while the simulation is running. That can be quite useful too, but it's only during the run. If you hit stop, you lose the things you just did. In this case, you would go to the contact plugin here. You'd open that up. Unfortunately, it doesn't show you the cell types right away. You have to open those up too. And then you can type the values. You type here 10. You type 20, remember 20.0, and hit return. So why don't people try that? I want everybody to see if you can go in, either by editing the base code or in the steering panel, go in and change the value from 10 to 20 and tell me what you see. And I'm going to run a little completion poll here just to make sure everybody's able to do that. While you're doing that, there was a question about what determines the timescales. Local movement is pretty easy, but reorganizing a large-scale object like the shape of the tissue as a whole means I have to move a lot of cells a long distance. You have to have a lot of cell-cell reorganization, and so that takes longer. The local boundary can relax almost immediately, but for the tissue to reorganize, you actually have to have cells move past each other, which takes time. There's a comment that the XML code has a run save option in Twidit, but not in Player. You can't edit the simulation code in Player. You can only edit the simulation code in Twidit. And so you can't save the simulation in Player. Does anybody want to put in the chat a picture of what the simulation looked like when they changed the contact energy? I really appreciate people having, there we go. That's great. I really appreciate people helping each other out in the chat. Again, your classmates are your best resource here for learning CompuSol. Great. Okay, I got something that looked like this when I increased the contact energy between medium and cells. And in fact, what's happening is the cells are getting pushed together more because they don't want to stick to medium. And if we were a little bit further along and could measure the total area of the aggregate, what you would see actually is the aggregate shrinks a little bit compared to what we had before with 10 and 10. All right. Okay. You may notice that the surface of the aggregate is smoother if you zoom in, and it also rounds up much faster than before. If you start the simulation again with 10-10, after a few hundred Monte Carlo steps, you'll see this. If you run it with 10 and 20, you'll see something like that. Higher energies produce higher forces. Velocities are proportional to force, and so the movement speeds are higher when forces and energies are higher. There's a question about which is cell, and cell medium. When you open up the plug-in here, contact, open up the energy terms. You see medium is 10. Medium, my cell is here 20. My cell is 10. They're all labeled. But you do have to open up all the little the little triangles to see it. If you edit it in the XML code, you see it line by line, it's written out. Why do you get a circle? Why don't we get a perfect circle? We don't get a perfect circle exactly because we're on a lattice. If we were off lattice, we would get a perfect circle. Here we see a little bit of alignment along the lattice directions. That's what I was talking about lattice anisotropy earlier. This isn't too bad. If it really looked like a square, you'd worry. Okay. Okay, now let's go the other way. Change medium and my cell to 5 instead of to 10. Are the cells going to stick to medium more or less? Is the aggregate going to become more compact or less compact? Think that one through and then make the change and tell me what you get. Somebody says less compact, more adhesion to medium. Give it a try. Okay. Does somebody want to put in the chat what you got? Yeah, some cells may separate. That's true. Ria, I think in your case, you must have changed something else. This is actually
cell parameter to higher, much higher than two times the cell medium. So
that's
separated.
fine. That's fine. So you're already jumping ahead and exploring more. That's great. That's always good to play. You're not going to break anything by changing those numbers, so you can explore them. Okay. If you make it 5, the adhesion is actually neutral. A few cells may separate, and you'll see that the boundary gets quite rough. If you look inside, you'll see that the cell-cell boundaries don't change if you've only changed cell medium energy. So the roughness between cell and cell is determined by the cell-cell contact energy. The contact with medium is determined by cell-medium contact energy. Now, make the contact energy between medium and my cell smaller, something less than 5. Make it 4 or 3. What happens? And Ria already basically did this exercise, so she's already seen that. Yes, when you restart it, it will always reset to 10 if you do it in the steering panel. That's why you might want to edit the source code instead. There we go. Yes. So you need to make the value less than 5. Yep. Great. So when you make the value less than 5, the cells stick to medium more than to each other, and they spread out. And that's a simple model of epithelial to mesenchymal transition. You've got a tissue that's become less cohesive to itself, or potentially more cohesive to medium, and the cells have spread out. Now, let's do one last exercise here, and then we'll jump to something else. We may skip a couple of these details, but because I want to get onto some other things. You can play around with this. Why don't you just try setting cell medium back to 10, and now set cell micelle energy to 1. What do you expect to happen? The cell medium contact energy is high, so the cells are repelled by medium. Cell-cell contact energy is low. So what do you expect to happen? Question, are the numbers relative or absolute? The configuration depends on the relative energies. However, the fluctuations depend on the ratio of these numbers to that thing we call temperature, to the fluctuation amplitude. And so if you multiply the cell medium, the J's, the contact energies, by 5, and the fluctuation amplitude, the temperature, by 5, everything stays the same. So if you make the cell-cell contact energy very small, the cells get very crumpled up. The cost of creating surface is small, and so the boundaries get very crumpled up. Right. Again, what really matters here are the relative values of the contact energies. Again, J cell medium versus J cell over 2, and the ratios with the fluctuations. So if you have contact energy that is very small compared to that fluctuation temperature, fluctuation amplitude, then you'll get very rough boundaries. You may even get single pixels that separate from the cells. If it's large, you'll get very smooth boundaries. And if it's too large, if the contact energy is too large compared to the fluctuations, you'll see the boundaries will line up on the lattice sites, lattice orientation. So here's an example. Here's J medium micelle equals J micelle equals 1. The boundaries with medium are very rough. The boundaries between cells are very rough. 10 and 10, they're not completely smooth, but you're also seeing that preferential alignment along the lattice orientations, which is something you don't want. 10-10 is about right. If you made it 20-20, you'd see things get pretty square. So why don't people play with that just a little bit? Try 1-1, 10-10, and maybe 20-20. Just take a minute to do that and see what the different morphologies are. People are getting all sorts of nice different shapes when they do that. Yes, if you make the cells separate by making the cell medium contact energy small, and then you make it big again, the cells will begin to cluster up. But if they spread out a lot, they may not find each other, so you get little blobs, little water droplets. Susan says that she's not able to change the values. Are you typing 10.0 into the editor? Okay. The volumes of the cells aren't equal. That's possible. Could one of the TAs please help Susan? Make sure that she's able to keep up. Okay, great. Katrin, what was the question about?
The question is, why is the volume different between the cells? How does this work? Because as far as I understood, we use the POTS model to have many pixels per cell, and we do not tell the cells to have a different volume. We just tell them to have a different energy to stick together or stick to the medium. And so I actually do not understand how they change their volume.
Okay, so
Why?
the cell volume should be pretty close for all of these. It's never exactly the same because we have a target volume, not an actual volume. The cells that if you make the contact energies with medium or between cell and cell, the cells will spread out a little bit because they're under less pressure. If you make the contact energy very large, the cells will shrink a little bit.
So this is already included in the model per default?
Yes.
Okay, thank you.
Gilberto, that's a great question, but I'm going to kick that one ahead because as soon as we're done with this lecture, Gilberto will explain exactly how that works. He'll run you through a bunch of exercises to do precisely that. So
Okay,
that's the next topic. Ramon, question.
Yeah. So when I decrease the cell-to-cell energy, I got after they just all spread out, they collapse again in small clusters. But I wanted all of them to go again to just one cluster, and I tried to increase the neighbor order. But what happened, it was that all the cells disappear, and I don't know if this is normal, but I don't.
Did you
understand it.
change the neighbor order in the code, or did you change it in the steering panel?
In the panel, in the player.
Okay, so I'm actually not sure that that works.
Okay.
In general, I've never tried doing that in the steering panel. It will work if you do it in the XML. But
Okay.
that one thing when you make the neighbor order larger, you have more neighbors, and you're summing over all of them. And we don't normalize by the number of possible neighbors. And so, in fact, using a contact energy of 10 with neighbor order 4 and is a bigger energy per unit area than a neighbor order of 2. And so there's a correction that we have to do if we want to change that neighbor order and keep everything quite the same. That's a little bit more of an advanced topic, and I'd like to talk about that later if we can. For today, let's flag that as something to discuss. But it would take maybe a little bit too much time to go into the details here. The mathematics, it's not a hard calculation, but it takes a little bit of math to do. And that's why we're not doing it in this lecture.
Okay, I get it. Thank you very much.
But Gilberto will talk about it also in the next lecture. Okay. All right. Again, the key issue here is this is a little bit of a mathy explanation, and I don't want to go into too much detail here about why the edges, the boundaries become rougher when you have a smaller contact energy. Essentially, the probability of moving the boundary is proportional to the inverse exponential of the energy cost divided by the fluctuation amplitude. If J medium is 10, then delta H is 10, and you're way out here, and so the probability of the move is low. If it's 1, it's here, and so the probability of the move is high. And if you change the fluctuation amplitude to be smaller, so instead of a fluctuation amplitude temperature of 10, you made it 1, then the probability of movements is going to be smaller. If you made it the other way, you would make it bigger. I'm not going to skip that. I'm going to skip that exercise now. People wanted homework. You can do that exercise as a homework problem if you like. 4-point function. But fundamentally, if the J the contact energies are 1 and the T is 1, that should give you essentially identical results to the contact energies being 10 and T equals 10, because it's always the ratio of those two things that matters. Okay. Now I would like to do a single cell simulation. We could do the single cell simulation quite easily by changing the code that we have here to start out with just drawing a single cell. Another possibility would be just to download the module 2._1_7 single cell that we have online. People who think that they can edit the XML to create a single cell to begin with, you'd feel free to do that, try it out. Otherwise, download module 2.1_7 and load that into Twidit and Player. Okay. Any questions on that? I'll give people a minute to do that. If you wanted to create a single cell in Twidit, you would edit the uniform initializer so it draws only one cell by changing the boundary boxes. It's easy to do, but it takes a little bit more time than I wanted to spend today. Gilberto will show you how to do that later. One of the problems, it's a little bit... please remember to mute your mics if you're not talking actively. Otherwise, we can have feedback. One of the little problems that we face, and it's again, we're learning a language here. And the language has vocabulary, it has grammar, it has syntax, is that everything needs everything else. But our learning is linear, and our presentation has to be linear. And so we have to do certain things first. And so when I present, for example, cell volume or cell adhesion first, contact energy first, there are going to be questions about how cell volume is controlled, how fluctuations work, and so on. If I presented it in a different order, people would start out asking questions about cell contact energy. And so I'm not trying to duck the issue when I say we're going to talk about it later, but we have to cycle back, and there are a certain number of basic concepts that we need to be able to start really understanding what's happening. And so I do ask for people to be a little bit patient as we cycle through these things. I'm happy to talk about them briefly, but a lot of these things we will address in the first day here, and I hope we can get things working. There's a biological question, which is how contact energy defined in biological terms. So cells have cell adhesion molecules, which are typically pretty specific. You have CAMs, you have cadherins, you have integrins, and many other cell adhesion molecule types. You also have things like sugars that have adhesive properties or repulsive properties. And so cells on their membranes have a whole variety of molecules that allow them to adhere specifically or non-specifically, or repel specifically or non-specifically other cells and cell types and extracellular structures. So, for example, integrins typically bind cells to extracellular matrix fibers. Certain kinds of sugars repel either other cell types or fluid medium, or actually increase affinity for fluid medium. And so cells have a whole variety of ways of adhering. Something that makes things a little bit more complicated in the biological reality is that the cell adhesion molecules reorganize And so the binding energy per cell adhesion molecule actually changes depending on the molecular organization. They often dimerize. Sometimes the dimers make pairs of dimers. And also, a lot of those cell adhesion molecules will then bind to the cytoskeleton, and that binding to the cytoskeleton can also change their affinity. And so in reality, the adhesion between two cells changes in time depending on how long the cells have been in contact and a variety of other things. Typically, we will neglect that. But if we really want to understand the details of cell adhesion, because it's important to our problem, then we can model that. Okay.
I guess
Okay.
just a quick add, I would say that most of the time in molecular biology, I've found that I don't ever consider the interaction between cells and their medium, but maybe that's more of a biophysical problem in, like, microfluidics or something.
Well, actually, the way cells behave depends extremely strongly on how they interact with the medium around them. You cannot understand how a cell behaves without understanding its context. I remember having a fight with a Nobel laureate once many years ago when I was young and daring, where he said if we understood the molecular species concentrations in every cell, this was pre-single cell omics, we'd know everything the cell was doing. The answer is it's hogwash. You could know every molecular species in a cell exactly, and you would have no idea what the cell is doing because it's going to depend on the cell context. Those molecules form fibers, there are motors inside of the cell. And so unless you know the relationship between the cell and its environment, knowing all of the molecular concentrations doesn't really tell you that much. There are certain molecular species that are very diagnostic. But for example, a molecule like caspase, some of the caspases, if those molecules are present but on the membrane, they promote cell survival. If they go to the nucleus, they promote apoptosis. And so where molecules are matters a lot, and their architecture matters a lot within the cells. And a lot of that molecular reorganization, which we're not going to be modeling here, depends on the environment the cell finds itself in. You can have a cell that's a frank cancer cell. You put it into a particular environment, it'll behave normally. You can have a cell that's a stem cell, you put it in the wrong environment, it behaves like a cancer cell. And so you really can't separate a cell's behavior from the environment it finds itself in. Again, there are exceptions to everything in biology, but in general, context is very important for cell behavior. Okay.
I would see that as chemical, not physical, though.
If I take an endothelial cell, a human endothelial cell, this is an experiment I used to do in my lab all the time, a human umbilical vein endothelial cell, and put it in a dish, and it's sitting there, and I take a little micropipette and poke it, it will start crawling. There's no time for any molecular synthesis, so the chemical composition of the cell doesn't change. The organization of the molecules within the cell change rapidly in response to the mechanical environment. And there are plenty of cells where stretching the cell and contracting the cell changes the cell's behavior. In the case of, say, stem cells, the differentiation of stem cells into different cell types is strongly dependent on the mechanical properties of the substrate. If I have stem cells and I put them on a soft substrate, they'll tend to follow neural fates. If I put them on a very hard substrate, they'll tend to follow bone morphogenetic fates. And so it's really very difficult. Sometimes you can separate chemical signals completely from mechanical signals and physical signals. So it's not impossible. And of course, there are electrical signals, which we haven't even talked about. But very often, the mechanical environment for cells is very important to what they do. And it's not that we should neglect the biology, the molecular signaling. Molecular signaling is very important, and often we will neglect the mechanical environment. But we should remember that we have to be careful when we do that. That's all. Okay, were people able to get that single cell simulation to work? Does everybody have something that looks like this? Does anybody need more time? Okay, there's a question about neighbor order in the plugins versus neighbor order in the pot simulation. Neighbor order in the pot simulation is about voxel updates. When I try to update and I pick the neighbors for the update rule that Julio talked about yesterday, that's the range. When I'm doing a calculation of energy, it uses the distance that I have within the plugin that I've defined. So you can have multiple interaction ranges defined for different functions in the system. That can be a little bit confusing, but it's a flexibility that can be helpful. Great. So it looks like people have a single cell. Does anybody need help with that? Okay. Let's set temperature to 10, volume to target volume to 1,000, lambda volume to 10, and J medium test cell to 10. Okay. And now either when you're running the simulation in the model editor or in the code, increase J medium test cell to 50. What happens? There's a question, can we have a non-homogeneous medium? I can take, instead of using the default medium, I can define something I call medium 2 and divide it into subcells. And I can have a non-homogeneous medium that way. And if I want to do explicit fluids, where I have the fluid flow and carry material and do advection, that's how I do it. We don't do it that often unless we're talking about solid extracellular matrix because it's computationally pretty expensive to do, but it works. And in the liver simulation I showed where we did explicit fluid flow in the sinusoid, we did it that way. So you can do explicit fluid flow that way. It's just computationally expensive. In the simulation, we're not going to see of cleft palate, but that has explicit extracellular matrix. Okay. So when you make J test cell 50, you'll see the cell will actually shrink a little bit and it'll get square. Okay, something like that. I'm going to skip the rest of this exercise. We're going to run out of time if I don't do that. This can be a nice homework problem. You can see what happens when you have very high contact energy. What I would like you to do before I do that, though, is there's one thing that is important. Why don't you make J medium cell very small? Make it 1 instead of 10. What happens? The biological, I don't like the term temperature. I regret using the word temperature 30-some years ago. That's a physics concept. Just think about the temperature as the cytoskeletally induced fluctuations of the cell membrane. And so the temperature is not the actual temperature of the system, room temperature, 37 degrees, whatever. The temperature represents the fluctuation amplitude due to cytoskeletal movement. And if I actually converted that cytoskeletal fluctuations into room temperature, real temperature, the fluctuation temperature of a cell is on the order of 10,000 degrees. Fluctuation amplitude to the cytoskeleton is much, much bigger than the actual thermal fluctuations in the cell because you have active movement of the cytoskeleton. And if you turn off actomyosin cytoskeletal movement using a molecule like cytochalasin D or blebistatin, you'll see the cell stop moving. Everything freezes up, it locks up. The tissues behave like liquids because of cytoskeletal fluctuations. Okay. Hayden would like to jump in and talk about the cell sorting simulation in just a second. Are you going to use this deck, Hayden, or do you want to use a different deck?
We can just use this deck.
Okay. So what happened when you changed the cell medium contact energy to be very small? Somebody show me a picture. It's allowed to be negative, by the way. Make the cell medium contact energy minus 5.0. What happens? Is that minus 5 or is that 1? Try minus 5. Yep. If you make the cell medium contact energy 0 or negative, there's no cost to creating new boundary. In fact, if you make it negative, you'll get what's called dust. The cell will fall apart because single point particles distributed in fluid have a lot of contact area. And you win by having more contact area. We haven't set a rule that says the cell has to be connected to each other. We haven't set a rule that the total area of the boundary has to be fixed. And so if we say that having a negative contact energy per unit area is a good thing, then the cell can fall apart. And this comes back to this idea that we talked about at the very beginning, which is in the real world, we take certain things for granted. Things don't just fall apart spontaneously. In a simulation, unless we assert something, the cell stays connected to itself. The cell has a fixed membrane area. Unless we actually assert that with an energy functional, it doesn't happen. And so the simulation is doing exactly what we asked it to do. It's trying to create as much surface area as possible, given the volume of the cell. And it can do that by having the cell fall apart into separated voxels. And so when you see these kinds of pathologies, you have to ask the question, did I forget something? Did I assume that there was something in my system that I didn't remember to put in by hand? All right. So I'm going to skip the details of calculating the neighbor order. This is how you correct for the contact energy as a function of the distance, because you should divide by the number of neighbors when you do change the distance. I'm not going to have time to do that today. But here's essentially what happens if I change the the radius of the contact area, excuse me, getting ahead of myself. If I change the interaction range for the same value of J, for 1, the effect of J is smaller than for 4 and then for 7. And so you'll see here the sort of stop sign where I have vertical line, vertical line, vertical line, 45-degree line. That means the fluctuations are too small relative to the energies. And I'm going to leave those exercises as ones for homework today, because I want to get to cell sorting. Okay, so Hayden, did you want to take over the lecture from this point?
Yeah, I can take over the screen share too. I've got the deck here.
Okay, great. I will stop the screen share and Hayden will take over for the rest of the lecture.
All right, so we are a little bit over time, and I want to make sure people have.
I moved the schedule to give us the extra half hour, so take your time here.
All right, so what time are we finishing?
We are going to finish at let's see, when did we start? I can't see what time it is now, so let's see.
It's 11:35 now.
Yeah, you have till 12.
Okay. All right, so it sounds like we're going to go to our first break a little bit later than usual today. So we do have a little bit of time to walk through this exercise. So to continue on with the topic of contact energy, we will actually walk through building one of the earliest CompuCell simulations of 2 cells sorting within medium. So I'll let this play to give you an idea of what the simulation looks like. So we looked at 1 cell aggregating and deaggregating in medium. Then we looked at single cell. Now we're going to look at cell sorting with 2 types of cell. And you can see here they started out in a random pattern, but due to differences in contact energy, they will actually sort out together, and you'll see that one engulfs the other. over time. So this is, we were talking a little bit before about kind of the biological basis behind this contact energy. This sort of sorting behavior is something that we see in nature during development, where in a randomly mixed aggregate of two embryonic cell types, they'll form clusters, one will engulf the other. And this behavior is sort of mechanistically predictable. So different cell types will engulf other cell types. And to the point where there's kind of a transitive property, even where if cell type A engulfs cell type B, and cell type B engulfs cell type C, then cell type A will engulf cell type C. So there is an underlying mechanism here that determines which cells engulf other cells. It's not necessarily unique to each individual cell. It's sort of by type. And like we were mentioning before, this is due to the different levels of cell adhesion molecules and the different cell types. So they have consistent hierarchy of adhesion energies per unit surface area when they come into contact with their own cell types and cells of other types. Kind of the key takeaway here, so this is from Steinberg's differential adhesion hypothesis. The idea ultimately in how we model the cells is that these values can be abstracted into a single scalar interface value between each cell type at the level that we are modeling the cells, which is kind of what we've been talking about already. And we'll talk a little bit about what that looks like in the code in the next coming cells, but to give maybe a closer look at how the contact energy is calculated with some simple examples here. So we've talked already about how the contact energy is sort of resolved into a matrix of cell values between each cell type. Again, what the underlying cellular POTS algorithm is trying to do is minimize the overall energy of the system. So we've already looked at how increasing contact energy between two cell types will cause those cells to seek other arrangements they don't want to touch if touching increases the overall energy of the system. So in this case, we have a checkerboard arrangement. We have the yellow squares represent cells in green medium. In this case, so you can see that we have 32 total interfaces between cells here. And each yellow cell has 4 exposed edges. So if we assume that our cells in this case are perfect squares, this is a situation in which energy is maximized because the cell differences here are occurring on every interface. So this is the most possible contact area between the cells in the medium here. So this would be a high energy state in this case. A low energy state would look more like a compact structure, where the cells minimize their contact with medium by grouping together. So this actually brings down the total number of interfaces between cell and medium to just 12 instead of 32. So this will reduce the energy. Therefore, the underlying algorithm will prefer arrangements like this, because again, we are reaching minimized energy in this case. If we introduce a second cell type, this is the same underlying process, so type B in blue here. But now we have, like we've been discussing, we'll have 5 energy values between cell A and cell A, cell A and cell B, cell A and cell medium, cell B and cell B, and cell B and medium. So again, same underlying mechanism, but it will involve a few more calculation variables. So again, we can represent the contact energy as an abstracted adhesivity value between each cell type, and the system will try to arrange into a pattern that minimizes the total amount of contact energy. So that's kind of why you get this engulfing behavior, because one cell will prefer interaction with the medium and another cell will not. And so in order to minimize the energy, it will try to, well, maybe try is the wrong word, but it will tend to group itself up in between the medium and the other cell that tends to prefer the medium. And we'll explore a couple of different arrangements here with our cell sorting simulation, which we will build next. So for those, I guess we do have time to actually walk through this. If you get stuck at some point, I want to leave enough time for people to explore the simulation. So if you do get stuck, or if you have problems building the simulation, we do have a completed version of it in the day 1 folder from yesterday, demo code. I'll go get a link to it here. So in the chat, I will put a drive link to sorry, let me label this. Yes, so it is important that everybody has this simulation. So again, I'll make sure that this goes out in our evening announcements email today too, a link to this since it's in the day 1 folder. We can make a copy in the day 2 folder too. But if we switch to, I'm going to switch my screen share to full screen here. Again, these instructions are in the slides, but we will go ahead and walk through actually building it together. So if you open Twedit, we're going to build a new simulation with Wizard. So we're going to go to CC3D project again, new CC3D project. We'll give it a name. We'll call it, I'm going to call mine Day 2 Cell Sorting 2024. You call it whatever you'd like. Again, simulation type Python plus XML. Go ahead and click next. In this case, so we're going to leave mostly everything the same here, but we want to shift from, we were using rectangular slab initializer before, we want to change that to blob initializer for this example. I believe, yes, we changed neighbor order to 2 as well. So again, change your pixel copy range or neighbor order to 2, and then your initial cell layout to blob. Everything else can remain the same. So then we'll click next. So in this case, we need 2 cell types. So we'll just put in, you can call them cell 1 and cell 2. In some of the literature around this, we call them condensing and non-condensing cells. So I'm going to use that. Condensing, non-condensing. So 2 cells plus medium. And then we'll click next. We're going to leave chemical fields blank here. So we'll just click next, and Wizard will leave it out of the simulation for us. And then here we need contact and, sorry, check my instructions again, and volume flex. So for your plugins, for this example, we're just going with contact and volume flex. And we'll click next and finish. We'll go ahead and open these up. And I'm going to throw up a completion poll once mine opens in Twedit. For some reason, my Twedit always has a hard time the first time I open a simulation for the day. All right, so I'm going to throw up a completion poll, see how many people have their simulation built and opened in Twedit. All right, looks pretty good. So looks like about half people have responded to the poll, but most of those people are completed. So again, if you need a little bit of extra time to get through, again, the instructions are in the slides, or if you would like to just grab the cell sorting code, I'll post it here in the chat again, so it doesn't get buried. You can grab the completed code from here and just open it in Player. Or you can open it in Tweeted and then open in Player. So once we've got our code open and Tweeted, I'm just going to go ahead and open in Player here. Again, it may take a minute on my end because I haven't opened anything yet today. So you can go ahead and open in Player, make sure that it runs. We'll take a look in a minute. So I'm actually going to do this without the rest of the slides for just a minute. These slides are available in both the deck for today, and then a more extensive, complete version of this activity is available in yesterday's module 1.4 content. So again, we'll send links out to that in this evening announcements tonight if you would like to take a look at the more extensive activity. But the instructions for how to build this are included in there, along with some questions for you to explore further on your own if you would like some homework. All right. So mine has opened. And you should be getting simulation that looks like this. So we have a blob of cells. The blob initializer creates and initializes cells in a circular pattern in the middle of the simulation instead of a rectangular pattern, which, generally speaking, is a little bit more biologically accurate. And your cells, I think, also start more circular in the blob initializer. I'm going to change my colors a little bit so that these stand out against each other a little bit more. Like this. Oh, that's a little migraine-y. Let me change to a darker. Oh, okay, that looks all right. So you'll notice that when we first start running our simulation, so we have a good question here. What decides how many cells we start with? We will take a look at that actually in just a second. We can look at the blob initializer. Yes, okay, James already answered that. So the blob initializer determines the number of cells based on the radius of the blob and then the width of each individual cell. So it fits as many cells of that width into the blob as it can. Somebody's cells disappeared. It's interesting. Like your simulation disappeared or the cells shrank and died? Anyway, while Masa gets back to us, you'll see that obviously the cells are not sorting as we showed in the beginning. And I think we've already had somebody answer in the chat why that is, and that's because all the interaction energies are the same, right? So if we take a look, let me go ahead and pause my simulation, switch back over to Tweedit, which is here. If we take a look at the actual XML code, so the contact energy specifications are handled by the XML. You'll see we have, so this is actually, there's a meaningful confusion in the chat from the previous exercises, I think, that I would like to point out here. There's some confusion in the double usage of the term type ID between the cell list and the contact energy plugin. So this is our cell type specification here. Again, we have 3 types: medium, condensing, and non-condensing. Medium type 0, condensing type 1, non-condensing type 2. If we come down to our contact energy plugin, which is here, plugin name equals contact, You'll see that this type flag is being used again. What this is referring to is the contact energy comparing two different cell types. I think that was a little bit confusing in the previous example because we only had two cell types, and type 1 and type 2 are being used for both types. You'll see in this case when we have three types that we're still only using type 1 and type 2 here. So cell type ID is different from the type flag that the contact energy plugin is using. This is just saying these are the two cells that we're comparing for each value in the contact energy equation. Hopefully that clarifies that a little bit. But so if we look at our contact plugin, we'll see that we have the same energy value for each cell interface type, which means that there's no sort of preferences between the cell types. There's no differential adhesion going on. They're all equally happy to stick to each other in the same patterns, which is reflected in our simulation by the cells not sorting. So to get us started, I'm going to pull the slides back up. So once you've found your contact energy plugin code block, we have, so you can go ahead and actually, in this case, you will need to stop your simulation. You don't have to stop it now, but we'll go ahead and stop it. Whenever you change values in your XML or Python files, you will need to stop and reinitialize your simulation. If you had paused it and resume it after you've made changes and saved, it will still be running the old code that's saved in your memory buffer. So whenever you change the underlying code files, you will have to stop and restart your simulation in Player. So if I'm going to double screen here briefly. I know that sometimes on smaller laptop screens, this makes this hard to see. I'll try to make the code larger here. And I'm going to take these values from our slide. So here we can make medium to medium contact energy 0. This value is in here because the contact plugin needs it, but medium to medium energy can be more or less anything. It doesn't actually have an effect on the simulation because medium is not an explicit cell type. So there is no actual cell interfacing going on between medium and medium. So we just set it to 0 just to represent that. And then we'll change our value from medium to condensing to 16. Medium to non-condensing to 16. Condensing to 16. And then we'll do condensing to non-condensing to 11. And non-condensing to 2. These values probably seem a little bit arbitrary right now. These are from the initial paper that was published using CompuCell to demonstrate cell sorting. We will manipulate these values in just a minute. So once we've made those changes, we want to go ahead and up here, click Save All. Now we've only made changes to this file, so technically you can just save this one. But in general, the safest way to make sure that you've updated everything is just to click Save All, and Twedit will actually save all files currently open in the editor for you. All of your files do have to be saved before running a new initialization of the program. All right, so now if we go back to Player, we've stopped our simulation, so now when we play it, it will reinitialize the files that we've just edited. So we'll go ahead and click Run. And you'll see now that our cells begin to sort. If you want to manipulate the file as you're going, we'll do what we were doing with our steering earlier. So if you drop down the contact plugin, you'll see you've got your energy values here, and you'll have to drop each one down to see which one's which. I believe these are in the same order as they are in the XML code. But this will give you an output, I believe, where eventually, given enough time, all of the non-condensing cells, in my case, the green, will aggregate in the middle and be engulfed by the blue. Yeah, Sharon makes a good point. I've made the non-condensing cells the one that condense. So I think I got my terminology backwards when I was setting the energy values. Or naming my cell types before. So again, if you open the provided code, those will have the correct terminology for condensing and non-condensing. Sorry about that. I should have named them in the opposite order when I was creating the simulation. But you'll see as it's running that the green cells begin to sort of join up into large aggregates and group within the blue cells. Yep, we got a good observation here. Some condensing types seem to be stuck in the middle of the non-condensing types, like bubbles. Yeah, so there's some issues here with, I see one cell type in the cell editor. Sharon makes the good point that if you're starting a new simulation, it's usually good just to close your player window and help clear out that cached memory. And then when you rerun from Twedit, it will load everything in. Sometimes if you've been running player for a long time, in a single session, it can kind of keep some bits and pieces of other code in the cached memory that can interfere, especially if you have simulations that are using the same variable names and things like that. So it's good just to quit player and reopen from Twedit. The initialization of the cluster created randomly, yes. So the initialization of the cells in the middle will create a randomized cluster of roughly 50-50, or if it's two cells, 50-50. There are ways of modifying that, and you can actually start with a specified initial configuration if you'd like. Jim Sluka will be giving a talk on that on Thursday, I believe, on loading cell configurations in from PIF files. But with the uniform and blob initializers, it does randomize the location of the cells. So I'm going to throw up one more completion poll here. Does everybody have the cell sorting simulation running, and are your cells beginning to sort? Yep. All right, looks pretty good. So we are running a little bit short on time now, but we thought it was important that everybody have a version of this simulation because it is a useful example of both the underlying POTS mechanism and the contact energy manipulations. And we do have an activity here that if people would like to do while we're at break, I'll leave it up on the screen for the next 10 minutes or so. You can modify the contact energy values to create a number of different configurations. So for example, the one that we're currently running, ultimately the goal is to get this engulfment configuration. But by manipulating your contact energies, you can get situations where cells split in half, roughly. Obviously, it won't look this clean, but you can get them to sort left and right. And it may not be left and right. It might be, because this is stochastic, it may be different directions, but you can have a line more or less down the center. You can swap the contact energies around to see if you can get the cell type engulfment to switch. So this is what Sharon was getting at. So right now, with our current configuration, if you followed me exactly, we have the condensing cells engulfing the non-condensing cells. I guess a good question to address next was how would you modify the contact energy values so that the opposite was true, that the condensing cells are engulfed by the non-condensing cells. And then you can also manipulate values to find this kind of more or less permanent and engulfed blobs configuration. which some people are already seeing a little bit of in the current simulation. Kimberly asked, by engulfing biologically, do you mean the neighbor influences the cell to phenotypically switch? No, in this case, engulfing just means that one cell type surrounds the other in the medium. So one cell type prefers to be in a cluster in the middle, and the other cell type prefers to be in a layer around the outside. And again, this is a 2D representation of a three-dimensional phenomenon. So in this case, you would have like a spheroid of one cells in the middle and then a sort of shell around the sphere on the outside. So good. Akshay has a good, his is approaching the engulfment, and obviously it won't be perfect in every case. You'll see that he's got a blue blob there in the middle. But Akshay's has approached engulfment. How is mine doing? Mine is not. Mine got stuck. So to save your current simulation, if it's currently running this way, it is already saved. You'll just have to locate where you saved it on your hard drive, and you can reopen it again later. And like I said, if you do lose it, we'll send out the link to the completed code later today. And there will be instructions in the slides on how to rebuild it. It's fairly simple. So I think I don't want to cut too much into our break. We will leave some time for Q&A here, though. I'll stick around. And if people want to work on this exercise, trying to get these different arrangements, I can leave this up on the screen for now. I think, James, if you want to launch the module experience questionnaire, if that's ready to go. Or I guess I can launch it too, since we don't have to save anything yet. Okay, good. All right, so we do have the poll up. So please do fill out the module experience questionnaire, and then we will. So we're going to come back at 12:15 then, James. If the schedule's been shifted. Yes, we'll come back at 12:15. So we'll take a 10-minute break here. And if people want to continue working on the cell sorting exercise and sharing their results, Shori shared a pretty cool one where there's a blob of cells in the middle with engulfment around the outside. Put this back up on the screen for people. You know, mine got engulfed blobs in mine. And again, the complete version of this activity in the day one folder has a few other stretch exercises you can do with manipulating cell shape, which we'll learn about in the next module, actually. So once we finish the modules for today, you should have the information you need to experiment with the other activities or the other prompts in the activity. We have one where I think you can add what happens if the cells are growing and dividing. What happens if you make your cells elongated while it's sorting? What happens if you add a third cell type? So there's a number of different things you can explore in that activity, which we'll send out for homework today. Optional homework, obviously. But this is a pretty fun simulation to explore.